So we're going to continue learning about cells. For some of you, again, it might be a review. For some of you, it might be brand new. And for some of you, you may look at it and go, oh, I vaguely remember this, but now it's time to relearn it. And we're going to go through this fairly quickly. I'm not going to spend hours and hours on cells because, again, you can do a whole entire PhD. You can spend your whole entire life looking at one type of cell. And we got to do all this in, a really quick time. So basically, cells are the basic structural and functional and biological unit of all known organisms. So we already talked about the characteristics of living organisms. So living organisms are made up of cells. Sometimes they're made up of only one cell, but they're made of this small unit that is its basic structure, its basic function, and the basic biological unit. So we call the cells the building blocks of life. All right, so a cell is the basic structural, functional, and biological unit of all known living organisms. And the smallest unit of life that is classified as a living thing and are often called the building blocks of life. Alrighty. So you should have watched the video earlier and hopefully filled in the chart. If you didn't fill in the chart, hopefully you just jotted down the names and kind of what they contributed to the cell theory. So when we talk about the cell theory, it's the basic three premises of what makes a cell, or where cells are found, that kind of thing. All right, so let's take a look at this chart. If you didn't get it while you were watching the video, no big deal. We're going to fill it in right now. And keep in mind, again, I'm going to say keep in mind a lot because I do. There's a lot of other people who contributed to the discovery of cells and the parts of the cells. We just have to narrow it down to what we can cover in a reasonable amount of time and kind of to some of the more famous people. And sometimes people are famous because they did discover something brand new, or they were very vocal about what they discovered, or they managed to write a paper first. So one of the first guys here, we have our Janssen guy who made the lenses, or was the eyeglass guy, We have Hook, who we don't know if this is what he really looks like since all his pictures were destroyed. We have Van Leeuwenhoek. Now his name is going to be spelled a whole pile of different ways. It's a Dutch name, so as long as you get it close to it, I'm good. We've got Schleiden here. All right, we've got Schwann here, and then we have our Russian guy, our Virchow here, and those other two guys there are our German guys. All right, so we've got our German guys, we've got our British guys, we've got our Russian guys. Alrighty, so our first guy, Janssen, he made a compound microscope. So if you remember, he took two lenses, put them together, and it really, really magnified together. Our Hook guy, he looked at thin pieces of cork. So just like the corks that come in wine bottles, we used to, when we had a lot more time and we used to teach about microscopes and cells from the very beginning, you didn't do it in elementary school, we used to actually have cork at school, and we cut it into little thin pieces, and you saw that they were made up of little boxes. This guy, Van Leeuwenhoek, and like we were talking about, you saw in the video, he looked at all sorts of stuff. You name it, he looked at it. Earwax, I can't even go into all the bodily things he looked at. He just looked at all sorts of stuff. Pond water, all that kind of stuff. All right, so he found animalcules, or he named the little moving creatures the animalcules. Remember, he's the first guy that kind of saw living creatures swimming around in something. Schleiden, this guy was studying plants in Germany. The Schwann, he studied animals. The way that I always remember, actually, was a student who mentioned it, and it's so smart. I always say it in my head. To know what the difference between these two people, what they discovered, since they do kind of, Schleiden and Schwann, they kind of have the S at the beginning of their name that can be confusing. The one student said to me years ago, oh, I remember Schwann did animals because Schwann sounds like swan, and a swan is an animal. So I'm like, oh my God, that's so smart. I've always remembered this. And this guy, Virchow, he looked at cells that were reproducing. Okay, so how did they contribute to the cell theory? So the cell theory in total, the first thing, Janssen, he allowed people to magnify objects. So people were going all over the place, taking their lenses and just looking at things under high power, which was really cool. If you figure back in the 1500s, people just looked at things through their eyeballs. They didn't realize that, blood flowed through veins. They thought there was 4 humors that made you happy or sad or sick or whatever it was. Robert Hooke, he coined the term cells, and remember he named them after the tiny boxes that the monks, like the cells or the little rooms that the monks lived in. Levon Hooke, he called these little things animalcules. We don't use that term anymore. Cells has stuck around, animalcules haven't, but it's still kind of cool when you see all his drawings. Schleiden, his was one of the parts of the cell theory that he said all plants are made of cells. Schwann, all animals are made up of cells, which to us is so obvious now, but again, we're going way back hundreds of years, and this guy said that all cells come from pre-existing cells. So because remember, way back when, they didn't have refrigeration, they didn't have the hygienic processes that we have now. So if you left something like a piece of meat out on a counter, and next thing you knew there were maggots falling all over it, people just thought that the meat turned into maggots when the meat was rotting. So he was showing that no, this new life has to come from life that was already there. He figured out that he watched cells actually divide from 1 into 2, 2 into 4. So here's our cell theory in a nutshell, the 3 points of the cell theory. So we've already talked about the first one. It's the basic structural, functional, and biological unit of living things, or living organisms. All right, all living organisms are made up of cells, which goes back to our characteristics of life, and all cells come from pre-existing cells. So magic doesn't just happen. All right, so let's take a look at the first type of cells that, or the simplest type of cells. All right, are prokaryote cells. Now, prokaryote, the term pro here, all right, means before. Kary means nucleus or nuclear material. So if you ever see the word kary, it's like a karyotype is all your chromosomes spread out, and you can look at it under the microscope and figure out if there's any genetic abnormalities in there, all right? So pro kind of means before nucleus was actually formed. So prokaryote cells, hugely important. So our cells can be prokaryote, and in this particular case, what cells mean is they do not have a true nucleus. So if you remember back to your plant and animal cells, which of course we're going to cover, we've got a nucleus, the control center of this cell. It's surrounded by a nuclear membrane. All the genetic material is safely packed in there. So these prokaryote cells don't have a true nucleus. They are simple cells with distinct features. So we're going to do a sample, or label the diagram of a sample prokaryote cell. Very interesting. If you're interested in microbiology at all, this again is one of those things you could spend your whole entire life studying one type of bacteria. So in our kingdoms, our bacteria and our archaea are the 2 kingdoms that are prokaryote. Now the other types of cells, so our plants, our animals, our fungus, and our protists are we call eukaryotic. And this U part here means true, right? And there's our nucleus name again. So these are organisms that have a true nucleus, and we'll talk about the true nucleus later, but basically it means that the genetic material is all put together in this nice membrane-bound nucleus. So these are our animal cells, our plant cells, our fungal cells, and our protist cells. So you should have a diagram. Again, if you don't have a diagram, just do your best job sketching one. We'll get you one in class. So this is kind of what a simple prokaryote cell looks like. Nothing what it really looks like in real life, because this is just drawn on PowerPoint. But we've got some distinguishing features here. So around the outside of this prokaryote cell. Now this is really important for how some antibiotics work, because some antibiotics can't get through this piece of, or this feature here. This is called the cell wall. So if you've got a bacterial infection and it's a type of bacteria that's got a really thick cell wall here, there's certain types of antibiotics that won't work for it. How other antibiotics work for it is they cause the cell wall to either not be formed properly, or the antibiotics will kind of punch holes in the cell wall, and the inside of the bacteria cell will kind of ooze out, and the whole bacteria cell will kind of disintegrate. So that's the cell wall. It's nice and thick. It protects the cell from things that might attack it. The cell wall also protects the cell from your white blood cells as well. So if you've got a type of bacteria that's got a really thick cell wall on it, the white blood cells are going to say, hey, there's a bacteria in here, let's go kill it. And this cell wall is nice, a nice wall of protection, so those white blood cells have to work really, really hard. In the cell wall, we've got our cell membrane. Now that cell membrane is really, really important. It allows certain things into the cell, such as water or glucose, because the cells need water and they need glucose. They need oxygen as well. Then it also allows things to leave. So things like carbon dioxide, which in high concentrations is toxic to a cell. Any other cell process, leftovers from cell processing can all get out of the cell. All right, and also the cell membrane just kind of is like a sack. It just holds everything inside. Now we've got the goopy stuff here, kind of looks like jello in the cell here. This is going to suspend all the organelles of the cell so that all the organelles aren't like, just drooping down here at the bottom of the cell. So this is called the cytoplasm. It's very, very watery, just like jello. Like it honestly is just like jello. It's got dissolved minerals in it. It's got dissolved sugars in it. It's got all sorts of stuff in there for the cell. It contains lots and lots of water as well. Now here, this squiggly little thing in here is the nucleoid. Now the nucleoid is genetic information. It's double-stranded DNA, which is deoxyribonucleic acid. Don't worry about writing that out right now. If you know DNA, I'm perfectly happy about that for the rest of your life. So this is, you can see here, there's no nice little membrane around here at all. It's just a little not circular, but little mingled up area of genetic material. Now another really cool thing that some bacteria have is this thing that's called a plasmid. This is an extra little loop of double-stranded DNA. And it is so important for us doing genetic engineering. Well, maybe not us, but some of us who will be doing genetic engineering. You can take a little piece of this plasmid and you can insert a gene in here, and this bacteria cell will actually start making the product that the gene codes for. So for example, a long time ago, if you were type 1 diabetic and you had to have insulin, you would have had to get insulin that's been isolated from a pig, and you would inject it into you. And a lot of times that causes problems because your body is getting something foreign to it. Now what we can do is we can take the gene, the human gene, from the human genome. We can insert it here in the plasmid, and this bacteria will actually make human insulin. So we can grow the bacteria in huge big vats in a lab. It can be producing insulin. We can take the insulin out of the vat, make sure we process it so it's nice and pure, and then give it to people so they can inject nice pure human insulin, which their bodies do not reject. So plasmids are really, really important. The other thing that plasmids are kind of a problem for, though, is it can also inherit the gene for antibiotic resistance, and then antibiotics that we take won't work against this bacteria because this bacteria will produce a product that destroys the antibiotic before it can work on that bacteria. Now, through here we have ribosomes as well, and you'll just see them scattered throughout the cytoplasm. The ribosomes, if you remember, or if you don't remember, that's no big deal, are really important for making proteins. The cells make proteins. What basically happens is the nuclear material codes, it gives the ribosomes the message of how to make the proteins, and then the protein, yeah, I think I said that right. What happens is the genetic material gives the instructions to the ribosomes, and then the ribosomes put the proteins together. I hope that made sense. I hope I didn't mess up too much there earlier. So the ribosomes are really important for making proteins. So another way antibiotics can work is they can go in and destroy the ribosomes. So the ribosomes can't make proteins, and if the bacteria cell can't make proteins, well, it's not going to live. It's going to end up dying. Now, how antibiotics work better for bacteria and don't kill off our own cells is bacteria ribosomes are just a little bit different than human ribosomes. So the antibiotics are made to recognize the bacteria ribosomes, but not the human ribosomes. So the bacteria ribosomes are the only ones that get destroyed. Now, not all bacteria have little flagella here. So these are little whip-like tails that can help the bacteria zip through watery environments. So if this was something like a cholera bacteria that lives in water, it would have these little flagella on it to help it whip through the water. Some have flagella, some don't have flagella on it. All right, it's just this is just a general prokaryote cell. And basically what the flagella do is it helps the bacteria move through the cell, or I mean through water. Whew, need a break from that slide. That was a long one. Alrighty, so the next cell we're going to look at here are our eukaryotic cells. So our eukaryote cells, like we said earlier, these were with cells with a membrane-bound nucleus. So the nuclear material is found in a nice little package with a membrane around the outside. The nuclear material is arranged in chromosomes. So you'll hear us talk about chromosomes, or you hear people talk about chromosomes. That's the genetic material, which is DNA, arranged into nice little groups of chromosomes. And you're going to learn a lot more about chromosomes when you take grade 11. We're going to talk a little bit about them when we do mitosis, but for now, let's just call the genetic material DNA, or chromosomes, or chromatin, whatever we want to call it. Eukaryote cells are a lot bigger than prokaryote cells, so we can actually see them under the microscope. Some bacteria you can see under the microscope as well, but we need to really stain the bacteria to see it, and you need to have a very good microscope. The school microscopes we have, some of them that aren't beat up too much, you actually can see bacteria. They're sort of unimpressive to look at under the microscope because they're just little dots of either red or purple, but they still are bacteria. It's still really cool to be able to see. So eukaryote cells are any cells that are in the animal, plant, fungus, protozoa, or algae groups. Alrighty, so you should have this diagram. Again, if you don't have it, just sketch it as we go along. I'm going to be babbling along as we go through here, so you can have a chance to sketch as I'm babbling. The other thing too is with the handout there comes a chart. So you can either just label this right now, just listen to me babbling, and then look up the chart, the parts of the chart later, or you can stop the video and fill out what the function of each of these things are as well. Or maybe you've got your notes from the past, or you've got a book with the stuff in it. However you want to do the function of it. I'm not going to spend a whole lot of time on the function because you'll be redoing this again in grade 11. And, I want to be able to get through this video within an hour. Alrighty, so this one is a plant cell. We're going to go through this again later, but for those of you, and if we were in class, I would say, oh, how can you tell this is a plant cell? And people would tell me the reasons of how they know it's a plant cell, but I'm just going to tell you all the answers. But if you want to, or if you can, try to think of the answers or think of the things before I say them, just to review yourself. And if you don't know anything, that's fine too. That's why we're doing this. Okay. So the first one here is the cell wall, right? So we've got cell wall in plant cells. Again, it's nice and thick. It's just like bacteria cells. So it's going to protect the cell from bugs and protect big, large things from getting in. It's made of cellulose. So this is our fiber in our diets. So this is what you're crunching on, and it's helping our food pass through. Inside that is the plasma membrane. Now it's the same thing as a cell membrane. We just call it plasma membrane for some reason when we're talking about eukaryote cells. Again, it's going to allow certain things in and certain things out. So oxygen, glucose are going to be allowed to come into the cell. Things like carbon dioxide, excess water are going to, or any processes, any chemicals from cell processes are going to move out of the water. This one here is called the Golgi body. Now, if you see it under the microscope, which is really hard to see under a light microscope, but if you see it under an electron microscope, the Golgi bodies are always described as looking like stacks of flattened pancakes. Now, the guy who first saw them, his last name was Golgi, so he named them after himself. So you may see this written as Golgi bodies, Golgi apparatus, or just Golgi. But it is with a capital G on here because it is named after somebody. So what ends up happening is the Golgi bodies package proteins. So, for example, for hormones. So they're going to take the basic hormone and they're going to package it into these little, we call them vesicles or vesicles, and they are going to be secreted out of the cell. Well, this is an animal cell, or a plant cell, so it's not going to be secreting as much stuff as human cells do. But anyway, so these are the Golgi bodies here. So they're going to package and process proteins. This next one, everybody always remembers this. This is the mitochondria. So the mighty mitochondria, it is the powerhouse of the cell. It's going to do the process of respiration. It's going to take glucose and oxygen and burn those two things together to form carbon dioxide and energy for us. It's going to release energy and carbon dioxide. And what was the other one I just said? Oh, and water, sorry. So it's going to produce carbon dioxide and water. So this hugely, hugely important for energy production for our cells. Our next one here is the nucleolus. All right, I'm having a little difficult seeing where that was going to. So this, in the nucleus, you're going to see this little dark spot. Hopefully when we get back and we're looking at the onion cells, you will be able to see the nucleolus. You always can see it. It's right in the center, not in the center, but it's always inside the nucleus. There can be one or two, sometimes three. So its job is to produce ribosomes. So that's its number one job. And remember, those ribosomes are really important because they produce proteins. The next thing here is we have the rough endoplasmic reticulum. So the endoplasmic reticulum is a network that passages or that substances can move through. So we always say it's like the transportation highway for the cell. So the rough endoplasmic reticulum looks rough because it's got ribosomes all lining the outside. And as the products get moved through this transportation package, the ribosomes are producing more proteins and sticking them on the outside. So it's kind of like finalizing a product. It's going down, like you're making a chocolate bar, you're starting off with your plain chocolate, and then we're having nuts and coconut and stuff like that added along. Gets to the Golgi bodies, and that's where it's packaged into the nice little packaging, and then it's pushed out of the cell, and then we eat the chocolate bar. I wish plant cells made chocolate bars. Okay, so the vacuole here is a great big storage bin of water. So plant cells are going to have one or two. It's usually really, really large. It's used to store a lot of water. It's the water that crunches out of an apple. When you crunch into an apple, you've broken open the vacuole and you're getting that big splash of water that's coming out. Now, also in this diagram here, this whole entire thing is the nucleus. This is kind of colored here to show you that the DNA is all arranged into chromosomes. So this is basically the control center of the cell. It's controlling the whole works of everything that goes into the cell. So when it's time to divide, when it's time to produce proteins, whatever it is, the nucleus is going to control that movement. Here's our little ribosomes that are scattered throughout the cell. Again, remember they're important for producing proteins. Next, we have our nuclear membrane here. All right, so that nuclear membrane is going to protect our nucleus. It's going to allow certain things in and allow certain things out as well. Our cytoplasm, same thing as in our prokaryote cell. It's a gel-like substance that's got a lot of water in it. It's got dissolved solids, dissolved sugars, dissolved salts in it. Now, here in our plant cell, we've got our chloroplasts, and you probably remember learning about them in the past. They're green colored, and these guys are really responsible for photosynthesis. So they're going to take carbon dioxide and water, and using the sun's energy, use it to produce glucose and oxygen. And of course, the glucose and oxygen we need, and we're going to use that glucose and oxygen, burn it in our mitochondria to produce carbon dioxide and water, which the plant cell is going to use to do photosynthesis. Alrighty, and then the last thing is the smooth endoplasmic reticulum. It's another transportation passage, but there's no ribosomes lining this one. All right, so that's our plant cell. We're going to talk about the differences between the two cells, the plant cell and the animal, but if you just kind of notice some of the differences, I've mentioned a few things already. We can, we'll take that up at the end. All right, so our animal cell, I'm not going to go through all the functions again, except for anything that's new. So for our animal cell, what do we have around the outside here? And you guys as a class would say, it's the cell or the plasma membrane. So you're correct. And next thing here, I'd say, what are these ones really important for energy for the cell? And you would tell me mitochondria. And then this guy here in the center of the nucleus is our nucleolus. We've got our rough endoplasmic reticulum around the outside. Again, it's got the ribosomes that are lining the transportation package. We've got our nucleus here, and we're showing again the genetic material wrapped up in or packaged up into chromosomes. We've got the nuclear membrane that's going to protect all that nuclear material in there. Now, around the outside here are they're barrel-like, they look like barrels. That's the description of them, is that they look like barrels, and they're at right angles to each other, and they're close to the nucleus. We call these the centrioles, or the two of them together is called the centrosome. Now, these are found in animal cells. We normally say they're not found in plant cells just for in grade 10 because it's just easier than trying to explain to you of where you can find them. You can find them in the lower plants, so such as lilies. All right, but the centrioles are absolutely essential for animal cells, and they play a big role in cell division. If you mess up these centrioles or mess up the function of those centrioles at all, the cell will not divide properly. So if we're talking about something like cancer, some of the anti-cancer drugs, what they do is they ruin the process that these centrioles do. All right, so they will, and cancer cells tend to divide really, really quickly. So what we're going to do is we're going to give the anti-cancer drugs, and they are going to get into the cells. They're going to destroy the process of these centrioles, and then the cancer cell can't divide. Now, it also, like I said, it happens to any of those cells that divide quickly. So things like your hair cells that divide quite quickly are also affected. So this is one of the reasons why your hair falls out, because we're also ruining the division of your hair cells. However, your hair does grow back afterwards. Okay, so we've got our cytoplasm again. We've got our Golgi body here, our Golgi bodies, and if this was like a sweat cell or a gland cell, it would be producing sweat or producing hormones. We've got our ribosomes that are there, really important for producing proteins, and we've got vacuoles. Now, we've got a lot of small vacuoles in an animal cell compared to a plant cell. Okay, so let's take a peek now at the differences between the plant. All right, so if we were looking at the differences between plant cells and animal cells in class, I would ask you, we would come up with some answers. It would take us an hour, but we're just going to bang through this really, really quickly. All right, so first thing is, plant cells have cell walls. Animal cells do not have cell walls. And every now and then I'll see a TV show or something, and the people in the characters will be talking about, I don't know, it's always a frozen human, and they're always trying to defrost the human, and there's always an argument about how quickly to defrost the human, and someone will say, you can't do it quickly because it'll break open the cell walls. So that's a blooper. So if you ever see that, that's a blooper there. Animal cells do not have cell walls. Animal cells have centrioles. We can call it a centrosome if it's two there, which they absolutely need to divide. In general, plant cells have a square-ish shape, where animal cells have a roundish shape. For grade 10, this is perfect for us. You'll see later on as you go through with more and more plant and animal cell biology how this isn't quite true in all cases. But for us, for now, perfect. Plants square, animals round. Plant cells have one great big vacuole, and animal cells have many small vacuoles. Plant cells have chloroplasts for sure. Animal cells will never have chloroplasts. A plant cell, because they've got that great big nucleus, that great big vacuole, the nucleus is pushed to the side. And a lot of times in animal cells, the nucleus will be in the center. Not necessarily true all the time, but plant cells for sure, the nucleus is mushed near the side. Where animal cells, the nucleus is pushed near the center. All right, so that's the end of this. Now, what I'm going to do with this is, what I'd like you to review first are the Quizlets. So I've got links to those on the calendar. And then once you're finished with the Quizlets, when you're pretty good and you think you know all this kind of stuff, go back and do the quizzes, because there's going to be 3 kind of topics all in one quiz. With all this, the prokaryote cell, the differences between plant and animal cells, and the cell theory, it's all squished into one great big quiz. You get 3 tries again, but there's 15 questions. I gave you a little bit more time. So again, your highest marks count. So take all 3 opportunities to do the quizzes. And it doesn't matter if you get, worse on the last one than you did on the first one, that's a-okay. Alrighty, so until next time, have fun. Go outside for a little while.